Hi, my name is Mortazavi. I'm an assistant in developmental and cell biology at UCI. My lab actually does a ton of different RNA-seq as part of all the functional genomics work that we do for the ENCODE project. But I am going to tell you nothing about that. Instead, I'm going to take you and tell you about two different projects we have in the lab. One which is about looking at time courses of development in these entomopathogenic worms. So that's going to be about 10 minutes. And then I'm going to spend another 10 minutes telling you about some single-cell RNA-seq work that we're doing as part of a European consortium. So I'm taking you essentially on both ends of the spectrum, right, from the least biomedically relevant to the most biomedically relevant. The common feature between the two parts is that it's really about trying to work with the lowest amount possible of RNA in every case. And these are both projects that are ongoing, so I'm not going to give you spectacular biological results, just a more proof of concept for some reason. Okay? All right. So why talk about nematodes other than C. elegans? I like to point out to people who are familiar with C. elegans that there are tens of thousands of other nematode species which have tremendous variety of differences in terms of their sizes, their niches, their life expectancy, their behavior. So, for example, something like 1 billion human beings are infected with these nematodes, and most people who are have no idea that they are. Yes, there's tremendous amount of conservation. So, for example, even though C. elegans is just 1 millimeter long, and Ascaris is about 40 centimeter long, they essentially share the same nervous system. And other parts are also highly conserved. So, my collaborators and I have spent many years actually sequencing many nematode genomes along the way. And the one I'm going to tell you about today is Steiner-Nema, carbocapsin, and a few of its friends. And these are entomopathogens. They're parasites of insects. So you can actually go and buy these nematodes, these parasites at Home Depot if you want. It's considered a normal and healthy form of biocontrol, right? For when you have insect pests. The idea being that the enemy of your enemy is your friend, right? So, say you've got this big bad pest, insect pest, and then these little infected juveniles find it. They actually come inside and they're going to essentially use a bacteria that they're carrying symbiotically to kill the host. And then they're just essentially going to be growing in there and replicating tremendously, and then come back out when they're ready. So it's actually an interesting way of studying parasitism while doing good for the environment. My colleagues and I, over the last few years, have sequenced a bunch of these Steiner-Nema. They have some reasonably sized genomes for nematodes. Their GC content is good, which helps for sequencing, and we have some decent assemblies. But I'm going to concentrate on comparing the transcriptome of this Steiner-Nema to this other one. Both of them happen to be some of the main ones that you can actually use for biocontrol. And we start asking questions in terms of, if we look at stage-specific expression, how conserved was it? And how did that compare when we looked at a much more distant organism like C. elegans? And so the short answer is about 25% of the one-to-one orthologs, the ones that are found in single copies across all 3 species, are actually showing conserved expression. Obviously, there's far better expression conservation between carbocapsin and Falcid than there is in elegans. And actually, we also found that, as you might expect, while it's less conserved, there's a subset of genes that are highly conserved in all 3. Along the way, doing this analysis for some other paper, we realized that we were having a real problem with the embryonic stages and getting the embryonic stages right. So even though it's easy to think of an embryo as a single stage when you're looking at the whole life cycle of an organism, in fact, even for something like C. elegans, you can actually subdivide the embryonic stages into many different subpoints. And in fact, a lot of work has gone into characterizing this and realizing that across species, closely related species, the time that it takes you to cross from one stage to another can take a lot. And so this becomes an issue when you're trying to match stages. So we were interested in trying that with Steiner-Nema. However, I was impatient. So I wanted to be able to do this a single embryo at a time. So the basic idea we had was, okay, well, let's first figure out if we could even do this. Let's go collect individual embryos of Steiner-Nema and then use this protocol that's in the literature for single-cell RNA-seq, use it for single embryo RNA. Okay? And we went ahead, and doing that, you can even see that, for example, when you look at multiple two-cell embryos, you can detect that these two-cell embryos have actually different RNA-seq expression patterns for a subset of their genes. And we've used that, in fact, to say that some of our two-cell embryos are going to be earlier and some of them are going to be later, based on the expression patterns that we're going to look at. We went ahead, and as I said, we broke this up into two different two-cell substages, and we believe that we could also do that with some of the other stages that we have. But essentially, we can now go and look at these clusters that we get in terms of our time course, and then go ahead, analyze them, and we can go through them that make perfect sense. And for example, actually, this is an early, these are the set of genes that are most highly expressed at 4 and 8 cell stages, right? And those tend to involve the mitotic cycle, stem cell maintenance, and so on. Those kind of things are being set up early in embryogenesis in nematodes. Whereas later on, we see that we actually first get neuronal differentiation to come up, and then later on, we get muscle differentiation. So, as I said, this was more of a proof of concept because now we want to be able to go back to the insect, dissect, get out the embryos from there, and then see how they're responding and not actually have to try to stage them like we did here. All right. So now for the second half of my talk. It often comes to surprise us to people that actually there is stellar heterogeneity in cells that otherwise are considered to be completely identical, say either from a clonal situation or not. Of course, many of you have heard it in specifications and so on, but the idea that there's stochasticity, that the transcription bursting is causing some cells to have higher amounts of RNA than others at different times, and that it's just a random process that averages out to turn out to be okay, is something that I think because of the tools we've used in the past, using large amounts of RNA, we can actually wash away. But if you were, for example, looking at it at single molecule level, you would actually be able to see that quite well. So I am part of a European consortium called SODEGRAPH, where we are actually looking at a particular subset of B-cell development and its connections with leukemias and so on. But essentially, we're working from this one particular B3 cell line, which represents the cyclin 3 B cells, that by simply increasing the amount of Icaros, transition to the next stage in their development, which are called these resting 3 B cells. The way that we're controlling that precisely is that we actually have an Icaros construct that has an estrogen receptor domain that's tamoxifen specific, so that when you actually add tamoxifen to these cells, you get the Icaros ER, you actually move into the nucleus and start the transition to the next developmental stage. My collaborators and I, this is a large consortium, have been collecting every omics data type you can think of. And so our original charge was to collect the DNA-seq in order to be able to look at open chromatin and changes in open chromatin during differentiation across this 24-hour time course. But along the way, a fluidine machine showed up on campus, and I jumped at the opportunity to actually use it in order to actually look at how the single-cell RNA-seq data compared to the pool RNA-seq data that we had collected earlier. So The big event was that actually at the beginning of this year, our Fluidine C1 machine showed up. And so the way the Fluidine machine works is that you essentially are going to be loading a suspension of live cells into this machine, and actually into this particular microfluidic cell. And then you are actually going to be able to isolate and then individual cells in this little mass, and then go ahead, lyse them, and then reverse transcribe and amplify the cDNA. And then after that, you extract the 96 cDNA libraries that you, well, the 96 cDNA samples that you have, and then you build those libraries which you can go ahead and use. We've gone ahead and done this, and so it turns out that our pre-B cells present some particular challenges because they're very small cells, and in fact, they're going to continue shrinking during this 24-hour time course. So they start being small, and then they become incredibly small. And that causes problems in terms of capture and building good libraries at the 24-hour time point, but in fact, at the beginning time points, they were pretty good. Now, what's happening is that these are the libraries that we built, and these are the ones that actually pass our filter. It turns out that only about 60% of our cells, after we've sequenced them, we decide to actually keep and analyze the data. But still, there's a ton of power out of having 324 of anything in order to do your analysis. Let me show you a little bit about what the profiles of these cells look like. So we've sequenced them on average to 1.25 million mappable reads. And what we found out is that in these particular cell types, the average cell is detecting about 4,000 to 5,000 genes. Now we've done different cell types by now, skeletal muscle and so on. And that average is dramatically different in those cells. So we believe that this is actually a feature of these B cells, not something that's specific to the technology. In the skeletal muscle cells, the average is about 8,000 transcripts per cell. All right. The kind of things that sort of are sobering, at least for me, when I look at this sort of data, is that in fact, there's only a handful of genes that we're detecting in every single cell that we're sequencing. It's less than a few hundred. In this case, it's 400. In the muscle cells, it's closer to 800. And in fact, there is a large number of genes that we're detecting only in a fraction of our cells. And we don't believe that this is an issue of sensitivity alone. All right, so we've done analysis of our data in several different ways. The main ones being self-organizing map, WGCNA, and the third one being a package called monoclone. I'm only going to tell you about WGCNA and monoclone, and I'm only going to tell it to you in terms of essentially sort of the behavior of the data, which is that we found out very early that our early time points are pretty uniform. So every cell in 0 hour is either, for example, for this set of genes going down, or for this set of genes going up. But when we look at our later time points, we're actually seeing more of a mixture, right? We're actually seeing that it looks like we've got two groups. For example, the ones that are actually going up here, and the ones that actually look like the original ones. We went ahead and we used another package that was published last year called Protocol for time course analysis. And the idea there is that we're going to take the cells and try to see if we can build a time course of the data where we allow for some of the cells to be actually delayed. Okay? So here we have, up here we have the 0 hour cells, and then there's a time course of differentiation here that was calculated by monoclone, and then we switch to the 2 hour cells, and then we get to 6 hours, then there's 18 hours here, and then 24. But in fact, monoclone calculated that there's some 24-hour cells that look more like 12-hour cells, right? It went back and based on looking at the behavior of the cells, decided that some of the cells did not look like they were as differentiated as they would be otherwise. So let me sort of describe to you the way we looked at the data before we had the single cell data, which was before this particular gene IGLL1 was a marker of differentiation, and it was actually highly expressed. So the little carrots here in the violet plot represents the average of the single cell, and that is actually what we measured before. And the averages match very nicely the behavior that we see now from the individual cells. But while we would measure an average, there were actually individual cells that were much more highly expressing this particular marker and some that were expressing it lowly. And when we got to 24 hours, most of the cells have turned off this particular marker as expected, but there's a few that still haven't. Looking at it in a different way, you can see that most of the cells that are early in this time course have it highly expressed. And the ones that are later in the time course actually have turned it down to 0. But there's still this whole additional population of cells sitting here that actually have not turned off this gene. And that is actually what's confusing the analysis when you're looking at it from the pool RNA, because you're still detecting some of the RNA from some individual laggards. We've gone ahead and we've actually looked at the behavior of many genes. The one that I'm going to focus on right now, because it explains also part of our problems with these cells as they're getting down the time course, is that they're turning on the RAG1 and RAG2 genes. So these are the genes that are involved in the DNA rearrangement. However, these particular B cells are lacking AX5, which is not going to allow them to actually progress and really do a B cell rearrangement. So in fact, we believe that the cells that are actually turning on RAG1 and RAG2 here are essentially causing massive chromosome instability that's going to kill these cells. So one of the reasons why we see these laggards at 24 hours, 8 hours is because those are the ones that have actually survived by not turning on RAG1 and RAG2 yet. Finally, the question I was really interested in, which is, what would you conclude if you were looking at the single-cell RNA-seq data for differentially expressed genes and then comparing it to the pooled RNA-seq data? For the sake of time, I will tell you that the pooled RNA-seq data detects thousands more genes that are being downregulated. But most of these are very lowly expressed genes. And most of them are just compatible with the ones that we detect both from the single cell data and the pooled data. The pooled data and the genes where they both agree happen to be the genes that everything else from our analysis is pointing out as key genes here. So everything that has to do with lymphocyte activation, all the genes that are involved in metabolic processes and so on, that are being downregulated during this time course where these cells are becoming resting and so on. All of those were detectable from the single cell RNA-seq data. Okay? So just to conclude, for nematode embryonic development, our goal is really to be able to see whether we could actually go and work from single embryos. No longer have to grow 1,000 embryos to extract their RNA and then do any analysis. Could we go down to single embryo? And the answer is yes. The methods are now, with SmartSeq2, are easy and straightforward, and we can build now dozens or hundreds of libraries as necessary for our studies. And in our case, this was just a proof of concept so that we could now start doing this across multiple species so that we could very precisely look at the conservation of gene expression, and just as interestingly, places where related species would show for orthologous genes a change in gene expression. We're interested in understanding that. On the single-cell RNA-seq side, the single RNA-seq is of such a nature that once you've actually seen the data, you never really go and say, no, I don't really want to know this. I want to go back to the life before, where I only had the pooled RNA-seq data. So it's very addictive. And it does help you find subpopulations in your cells. So some of you presumably have thought, just like me, that there have to be there. And it's one very powerful way to get there. And in our case, we're really interested in looking at being able to characterize the subpopulations in our cells in order to be able to make sense of the chromatin data we have, where we see a lot of gene changes at the level of RNA, and we don't see as many changes at the level of the chromatin. One explanation is it's really just a subpopulation of the cells that are changing the expression of their genes, and that as long as the majority of the cells are not changing, that's what the chromatin data is going to be measuring, right? It's going to be measuring the major population. If it's only the minor one that's changing, that would explain the disconnect that we see often between RNA-seq and open chromatin. All right, so all the single-cell RNA-seq work that I told you was the work of my graduate student Ricardo and my lab manager Nicole, with some assist from other members of the lab, and some of our collaborators as part of this EU FP7 project. And then the work that I told you about the single-cell embryos in the nematodes, that's the work of Marissa along with Christy. And this is as part of a much larger collaboration I have with my one of my long-term collaborators, Adler Dillman, who's now at UC Riverside, Paul Sternberg, my old postdoctoral advisor, and this consortium of people who helped us with sequencing the Steiner-Niemann genomes. Any questions?
You said that in your 24 hours window, there was a selection for survival.
Yes.
How do you select the dead cells actually weeded out?
Well, so the cell, as I said, the cells are shrinking.
Yeah.
And it also looks like the ones that are going to turn on RAC1 and RAC2 prematurely are probably dying. And so I think that when we look at the cells we have left after 24 hours, we've just increased the ratio of the ones that actually did not respond. So we do see more cell death, for example. I mean, we do see shrink. We see cells that have shrunk and so on.
You don't sort.
We're not sorting them. One of the problems we could try to sort them on something. Turns out these cells are all getting out of the cell cycle, so we can't even sort them on whether they're dividing or not. The other thing is we do stain them before we use them on the C1. Well, as I said, we captured 167 cells. But by the time we looked at how many of those cells still had passed a lot of our filtering and quality criteria, we were down to like 67. Right?
So this is like excluding ones that.
So we captured 142 cells that on the C1 looked good. Right. So a C1 can capture right now 96 cells. So they can capture up to 96 cells. In our case, we did a first run and had few enough cells below the number we wanted, so we did a second run. And with the second run, we didn't actually increase the ratio of cells we were recovering. So, and our filters are, so another thing we're seeing is that there's cells where we capture them and we get libraries, we get a lot of reads, but the reads do not map onto the transcriptome. And so we believe those could be cells where we're in the process of dying and where somehow we're getting a lot more DNA into the mix somehow. And that's what's causing the low mapping rates onto the transcriptome. So one of the filters we have is that you have to have a minimum 50% mapping rate onto the transcriptome for us to even have it into the analysis I showed you, downstream analysis. Yes?
So you said that you think I think the next one showed that although there's an average of 5,000 per cell, only 200 or so are shared.
I think 300.
You know, how do you realize this is low sampling? There's a lot more expression. So how do you realize that this is just
well, so it's based on looking at first of all, we have some spike-in sets that are included so that we can see, for example, what are the number of transcripts that should be in the cell based on the ERCC spike-ins we're putting in. Right? So, but The other thing is we can look at the profile of what locally expressed genes look like versus the ones that are bimodal. And so, for example, when you have situations like this, where most of the cells are not expressing, but you have a few that are extremely highly expressing, that is a very different situation than when you actually have cells that are mostly not expressing and a few. The scales are actually not the same, I'm sorry. So this is 0 to 100, this is 0 to 10, right? So when you have this situation, we see that the subsets, the distance between what we have to detect and not detect is high enough that we don't expect that it's above capture rate. The speculation within the field is that the true capture rate of single molecules is somewhere between 10% and 50%. So the prediction would be that if you're actually there are more than 10 transcripts per cell, most of the time you should be detected. As I said, when we looked at the single cell data that we have for myoblasts, as opposed to these red T cells, this average has shifted by like 3,000 over. Now those happen to be also bigger cells.
What percent are shared in that situation in myoblasts?
Not that much more. This doubles maybe. That's about it. And in fact, there's many genes involved in myoblasts identity that are actually not shared by more than a handful of times. Right? I didn't show you because I didn't put it on, but Icaros is one of these key identity genes for B cells, EGF1 and the others. They're all detectable in most cells at healthy levels. So genes that were expressed at healthy levels in the pooled RNA-seq data are showing up at healthy levels here too. And the averages that we're calculating are matching the averages that we want to measure.
Help me understand the biological significance of this biomodality. Are these just cells that haven't gotten the memo yet and they're going to catch up a little later? Or are they going to get cold because they're sort of lying around and didn't get dizzy? Or
well, okay, so in the case of B cells, you can think of B cell development or T cell development and so on as all going through different funnels where only subset of a particular genes will come out of them. Right? In the particular case here, these cells need to go into this resting stage right before they start doing their B cell receptor rearrangement. Now, why do they have to go through this resting stage where they stop all metabolic activity in order to do the rearrangement? I'm not sure. Maybe it's something about stability at the time when they're going to go and cut their own DNA. But most cells will not make it through this, right? Whether it's because they will die between here and here, or they will die after they do their rearrangement and the rearrangement fails somehow. I do not know. But the idea that cells that start from a uniform field of identical cells end up responding to their macroenvironment, or they start responding to interactions with neighboring cells in such a way that they start changing their programming. Something we've all known about in particular subsets of mutations. So my sense is that cells just have to go to respond. Now, cells that are being treated in a dish with lots of them may not be responding the same way as they would be in the body of the mouse. Obviously, we're hammering up to get this and that. But it does actually reflect what these cells go through. They just simply increase their level of Icaros. So Icaros is already here, expressed at some level, which is simply shifting them on the Icaros. That's enough for them to shift from one stage